Okay, so today we're diving into something pretty complex and potentially impactful.
Definitely a big one.
Yeah, it's the proposed release of genetically engineered mosquitoes,
Hmm.
specifically the Aedes aegypti mosquitoes, the OX5034 strain, in Queensland, Australia.
Right.
We've got a stack of scientific commentary and reviews here,
Yeah.
and a lot of them, well, they've raised some pretty serious concerns about this whole plan.
A lot of red flags.
And what we want to do is really pull out the crucial insights from all of this, help you understand what these experts are really worried
Yeah,
about.
exactly. We want to go beyond the headlines, really look at the ripple effects these releases could have.
Yeah.
Like one of the first things that jumps out from these documents is the possibility of more allergic reactions.
Okay.
And, you might think, oh, mosquito bites, itchy, annoying, but it could be a lot more serious than that.
Okay, yeah, let's break that down. I mean, mosquitoes are annoying enough as it is.
Huh.
But the idea that this could lead to, actual allergic problems is definitely concerning. What are the sources saying about that?
Well, what's interesting to think about first is just the Aegypti mosquito itself
Okay.
and how it behaves naturally.
Right.
Unlike some other species, they tend to live indoors.
Hmm.
And here's the thing, even the males are attracted to humans.
Hmm.
So even without any genetic modification, just the fact that they're hanging out in our homes means we're exposed to their allergens more often.
So it's not even just about getting bitten, just having them around inside could be a problem for allergies.
Exactly. And then you introduce the genetically engineered OX5034 males into the mix.
Right.
These guys have novel proteins. That's the key point here.
Yeah.
And those proteins, well, they share very similar sequences to proteins that we already know can trigger allergies in people.
Oh, wow.
Think of it like, if your body reacts to a certain shape of protein, let's say in birch pollen, a very similar shape in these mosquito proteins could also kind of trick your immune system into having an allergic reaction.
Oh, that makes sense. So someone who wasn't even particularly allergic to regular mosquitoes could suddenly develop a problem.
That's a real concern these documents raise, like the potential for brand new allergies or for existing allergies to get much worse. And it's not just about breathing in allergens either.
Right.
The sources also talk about a potential increase in skin reactions.
Okay.
While mosquito saliva itself can cause irritation, like we all know,
Yeah.
direct contact with these novel proteins, maybe by accidentally squashing a male mosquito, could also trigger a response.
So it's like multiple ways you could be exposed. And I noticed the documents also mention something about competition for food playing a role here.
They do. Male mosquitoes, they mainly feed on nectar.
Right.
But if you release millions of these OX5034 males, well, they're going to be competing for that same food source.
Yeah, of course.
This could force them to look for other options. And for Aedes aegypti, that can mean trying to feed on human blood, even though it's not their preferred diet. More males potentially biting could definitely lead to more allergic reactions.
That's interesting. So even though the goal is to reduce the mosquito population in the long run, this initial release of engineered males could actually increase the overall number of Aedes aegypti in the short term. How does that impact the whole allergy situation?
Right, more mosquitoes around, even if they're mostly males, means more chances of being exposed to allergens generally.
Yeah.
Studies have actually shown that even in well-sealed homes, you can find traces of Aedes aegypti in the dust,
Wow, really?
and that dust contains allergens. Releasing more males, it's been shown to actually increase the number of male mosquitoes found inside houses.
No kidding.
Plus, the pheromones that these released males give off can attract even more mosquitoes, both male and female, into our living spaces.
So it's kind of like an unintended consequence, You're trying to reduce the population, but in the beginning you might actually make the allergy problem worse. And isn't there a chance that the genetic modification itself could have an impact on the allergens that are already present in the mosquito?
Absolutely, that's another big concern. The process of genetic engineering, it could accidentally alter the levels of the naturally occurring allergens in the mosquito compared to the wild population.
I see.
We just don't have enough data on that yet.
Yeah.
And going back to those specific novel proteins we talked about, the documents highlight these sequences, SF-PEGF, DGG-VAT, GGV-ATV, and TIGR.
Okay.
And they really emphasize that these bear a striking resemblance to known allergens, like BET-V1, which is found in birch pollen,
Mm-hmm.
and SOLA-T1 from tomatoes. That molecular similarity, that's a major red flag for the allergy concerns.
So just to sum up this whole allergy issue for our listener, it sounds like we could be looking at a double whammy here. More exposure to allergens from a temporarily larger male population and potentially more potent allergens within those mosquitoes because of the genetic modifications.
Exactly.
And it's not just bites, it's breathing them in, it's skin contact. It's a lot to think about. Now, these documents also raise some serious concerns about disease transmission.
Yeah, this is a huge area of concern. The release of OX5034, it could actually increase the risk of diseases like Zika and dengue in some ways.
Really? How so?
One interesting point the documents bring up is that a higher density of male mosquitoes can actually lead to more frequent interruptions during mating.
Okay.
And this can result in female mosquitoes mating with multiple males. It's called polyandry.
Uh-huh.
And the sources suggest that this could, kind of paradoxically, increase disease spread.
Wait, so more mating could lead to more disease? That seems a little counterintuitive if you're trying to reduce the number of offspring, right?
Right, it does seem a bit strange. The exact mechanisms, they're still being studied, but it could be something about the increased contact rates between infected and uninfected mosquitoes.
I see.
Like, if an infected female mates with multiple males, she could potentially spread the virus to more of them.
That makes sense.
There also might be some changes in the female's behavior or physiology after multiple matings that could affect how the disease is transmitted.
Interesting. And then there's the whole issue of female OX5034 mosquitoes surviving when they're not supposed to. The whole plan hinges on them dying off, right?
Absolutely. The self-limiting trait in these mosquitoes, it's designed to prevent female offspring from surviving,
Right.
but the documents point out a couple of major ways this could fail.
Okay.
First, if any female OX53D4 are released accidentally, which is always a possibility in large-scale operations,
Sure.
that immediately increases the risk of biting.
Yeah, of course.
Second, the self-limiting trait, it's suppressed by the presence of tetracycline,
Okay.
which is an antibiotic. If the larvae are exposed to tetracycline in the environment, and the documents mention potential sources like animal feed or contaminated water, the female offspring could survive to adulthood and potentially transmit diseases.
So tetracycline seems like a key vulnerability in this whole system.
Definitely. And the documents highlight other ways that this release could accidentally increase disease risk.
Okay. Like what?
For example, if the released male mosquitoes are too good at competing for nectar,
Right?
which is their main food source,
Mm-hmm.
they might start looking for blood meals from humans more often.
Makes sense.
And similarly, female mosquitoes that don't get enough sugar from nectar, they're actually more likely to transmit Zika virus.
Oh, wow.
So ecological disruptions caused by the release could actually make the remaining mosquitoes more dangerous.
That's a pretty concerning feedback loop. And it gets even more direct, right? Because the released males themselves could become carriers of disease.
Exactly. This is a huge point. Male Aedes aegypti, they can actually acquire viruses like Zika from infected females through venereal transmission
Oh, so during mating.
during mating. And studies have shown that this can be a surprisingly efficient route of transmission, almost as effective as a female getting infected through a blood meal and then biting someone.
So the very mosquitoes that are intended to reduce the population could become disease vectors themselves and spread those diseases to the wild female population.
It's a real possibility, especially when you consider how frequently male Aedes aegypti can mate.
Yeah, how often are we talking?
Some sources say up to 30 times a day.
Wow.
Releasing millions of these males, it just dramatically increases the chances for this venereal transmission to happen and amplify disease spread within the mosquito population.
That's pretty alarming. And the documents also mention that even the offspring that are supposed to die off could still play a role in disease transmission.
That's right. They talk about the risks of vertical transmission,
Okay.
where an infected parent passes the virus to its offspring,
Right.
and trends of aerial transmission,
Okay.
where the virus is actually present inside the egg itself. So even if the first generation of hybrid offspring have shorter lifespans, they could still be born already carrying a disease.
That's a scary thought. And it's not just through bites either. The documents mention other ways these viruses can spread between mosquitoes.
Exactly. Both Zika and dengue, they can spread through horizontal contact transmission, meaning just direct contact between infected and uninfected mosquitoes, whether they're male, female, or even larvae.
Wow.
This can happen through contact with infected urine and feces in breeding sites, leading to more transmission through eggs and direct contact. There's even a mention in one of the sources about male Aedes aegypti exhibiting non-heterosexual behavior, which, well, that could potentially contribute to disease spread between males through this kind of contact, particularly in areas with dense mosquito populations.
It seems like we're just uncovering more and more layers of potential unintended consequences here.
Yeah. It's a lot to consider. And then we also have to think about the lifespan of the released OX5034 males. The documents say they can live for several weeks, which is definitely long enough to potentially acquire and transmit diseases.
Mm.
And importantly, we don't actually have specific data on their survival rates in the real environmental conditions of Queensland.
And what about the genetic modifications themselves? Could those have any unforeseen effects on the mosquito's ability to carry or transmit diseases?
That's another area where the documents express concern. Certain proteins in the male accessory glands of OX5034 mosquitoes, which can affect female physiology and their gut bacteria,
Right.
those haven't been thoroughly evaluated for their potential impact on disease transmission.
I see.
And similar changes in salivary proteins or odorant binding proteins in these modified mosquitoes could also pose unknown risks. Even the seminal fluid from OX5034 males, that might alter how female mosquitoes search for hosts, how much blood they take, how long they live, and whether they mate again. All of which could have implications for how diseases spread. And if females end up feeding on multiple hosts because of any of these changes, that could also spread diseases mechanically.
So this initial increase in the male population, even if it's only temporary, could create a window of higher disease risk because of the increased mating and the potential for venereal transmission.
That's a key takeaway. More males initially means more mated females. And mated females tend to live longer and take larger blood meals,
Right.
both factors that increase the likelihood of disease transmission. Plus, more competition for egg-laying sites among these more abundant mated females, that could actually increase the risk of passing diseases to their offspring through the eggs.
Oh, wow.
And it could even lead to larvae with nutritional deficiencies, which might make them more effective disease vectors later in life.
So even if the program succeeds in reducing the overall female population, the disease risk might not actually disappear.
Exactly, because Aedes aegypti, they really prefer human blood. A smaller female population might end up feeding on humans even more often, increasing the transmission rate to us, even if there are fewer mosquitoes overall. And we can't forget about the possibility of hybrid vigor in the offspring of OX5034 males and wild females.
Right.
These hybrids could potentially live longer and have more opportunities to transmit diseases over their lifespan.
That's a lot to consider. Compared to other mosquito control methods like introducing Wolbachia bacteria, it sounds like OX5034 might have some inherent disadvantages when it comes to disease risk.
The documents definitely suggest that. For instance, Wolbachia infection, it can actually inhibit the replication of certain viruses inside the mosquito. OX5034 doesn't have that kind of built-in safeguard. And the use of antibiotics in rearing OX5034, that raises the worry of antibiotic-resistant bacteria developing in the mosquitoes, which could potentially even increase the risk of dengue transmission.
Yeah, antibiotic resistance is a huge global health challenge as it is. It's definitely something we don't want to make worse. Now, let's shift gears a bit and talk about some of the ecological and ethical concerns that these documents highlight.
Yeah, absolutely. One of the major ecological concerns is that the release of OX5034 could lead to the Aedes aegypti population being displaced by another mosquito species, Aedes albopictus.
Oh, wow.
We've actually seen this happen in other parts of the world,
Okay.
where Aedes albopictus has become the dominant species after Aedes aegypti populations declined for various reasons. If this happens in Queensland, we could end up with two different disease vectors coexisting, which could potentially increase both allergic reactions and the overall burden of mosquito-borne illnesses.
So an intervention that's aimed at one problem could accidentally create another one that's potentially worse.
Precisely. And going back to that antibiotic issue, the use of tetracyclines in the breeding process for OX5034, that could contribute to the development of antibiotic resistance in the gut bacteria of these mosquitoes. There's even some evidence in the document suggesting a link between Oxitec releases in other regions and increased tetracycline resistance. Studies have found chlortetracycline-resistant bacteria in the genetically engineered larvae.
So this isn't just a problem for mosquito control, it has much broader implications for public health in general.
Exactly.
What about the impact on other wildlife in Queensland?
The documents are pretty critical of the lack of research into how OX5034 might affect other organisms that interact with Aedes aegypti. This includes predators like various insects, amphibians, reptiles, birds, and mammals that might eat these mosquitoes. There's a real lack of long-term studies looking at the potential effects across multiple generations for these animals, many of which play important roles in the Queensland ecosystem.
And Queensland has a particularly diverse range of species, including many that are endangered, right?
It does. For example, all those millions of released male mosquitoes will be competing for nectar,
Right.
which could negatively impact native pollinator populations, including endangered butterfly species that rely on nectar for survival. Tadpoles, which are natural predators of Aedes aegypti eggs and larvae, they could be harmed by eating the transgenic proteins and any heavy metals that might be present in the OX5034 mosquitoes. A lot of amphibian species in Queensland are already facing extinction.
So it's a delicate balance, and introducing a new element like this could have unforeseen consequences up and down the food chain.
Absolutely. Reptiles like geckos and water dragons that eat Aedes aegypti, they could also be affected, potentially impacting endangered reptile species. Birds, even chickens in Queensland, they eat mosquitoes, and the risks to endangered bird populations haven't been properly studied. Even smaller predators like copepods, crayfish, flatworms, and arachnids, all of which prey on mosquito larvae, could be harmed.
Wow.
This could disrupt natural mosquito control mechanisms and potentially affect endangered crayfish species. And we can't forget about carnivorous plants that trap mosquito larvae. They could also be at risk from eating these modified mosquitoes.
And the possibility of these modified proteins and heavy metals accumulating in the food chain is another serious concern, right?
Absolutely. Bioaccumulation in predators is a real worry. It could potentially pose risks to animals higher up the food web, including humans who might consume these predators. Heavy metals can also be absorbed by algae and other organisms, entering the food web at a very basic level. The documents are very critical of the toxicity studies done by Oxitec.
Okay.
They say they're too short and too narrow in scope to really assess these long-term ecological effects.
It sounds like there's a lot of uncertainty about where these genetically engineered mosquitoes might end up and what they might interact with in the Queensland environment.
Yeah, and geographic containment is another issue raised in the documents. The fact that the OX5034 production facility is in the UK, which is within the flight range of Aedes aegypti,
Right.
and that the mosquito eggs can survive even freezing temperatures, that raises questions about accidental escapes.
Right.
And the self-limiting trait itself, it relies entirely on the absence of tetracycline. If the mosquitoes spread to areas where tetracycline is present, the female offspring could survive and reproduce.
And that would completely defeat the purpose, wouldn't it?
Exactly.
What about the ethical considerations here and the views of the people in Queensland and beyond?
Public opinion is definitely a big factor. The documents mention a large online petition with signatures from all over the world, expressing concerns about the release and highlighting what they see as a lack of consent from the community. Given the potential for widespread ecological and public health impacts, the ethical implications of going ahead without broad public support are significant.
It really feels like there are so many unanswered questions about the long-term safety and the ecological consequences of this release.
It does. The documents really stress the lack of health studies on all the different predators in Queensland that might eat OX5034,
Right.
or on the effects of eating prey that have consumed these modified mosquitoes. And the evidence for human safety through direct or indirect consumption is basically non-existent.
So a lot more research is needed.
Definitely. And one of the most far-reaching concerns is the potential for gene transfer.
Okay, what does that mean?
Well, there's a risk that the transgenes from OX5034 could be transferred to the gut bacteria of insectivores, humans, even to bacteria in the soil or water.
Wow.
The piggyback transposable element used in OX5044 that hasn't been fully evaluated for its ability to move around and spread genes, potentially with the help of other naturally occurring transposable elements. And studies have shown that DNA from genetically engineered organisms can actually become part of the microbiota of animals.
Wow.
And transgenes from genetically modified foods can survive digestion and potentially even transform bacteria.
Really?
So this is a major area of risk that hasn't been adequately assessed.
Wow, this has been an incredibly detailed look at a very complex issue. So to bring it all together for you, our listener, what are the main interconnected risks that these documents highlight?
Well, we're looking at a whole bunch of potential problems here. First, there's the risk of increased allergic reactions because of the novel proteins and the changes in mosquito behavior.
Right.
Second, there's a higher chance of disease transmission through various pathways, which could actually be made worse by the release itself.
Okay.
Third, there's the potential for significant ecological disruptions, including one disease vector being replaced by another,
Right.
and harm to a wide range of animals and plants, including endangered species.
And finally, there are those big unanswered questions about antibiotic resistance and the potential for gene transfer.
Exactly. It's a lot to be concerned about.
So what's the main message from these sources about how to move forward, if at all?
The overwhelming message is that we need a lot more research. comprehensive, independent, and long-term safety studies that are done specifically in the environmental conditions of Queensland before any releases are even considered.
So they're saying the current data isn't good
They're
enough.
saying it's insufficient. It's often based on lab studies in mosquito populations that aren't even from Queensland. It's lacking the in-depth investigation needed to really understand the full range of impacts we've talked about.
It really highlights how complex natural systems are and the potential for unintended consequences when we introduce genetically modified organisms into the environment.
Definitely. It calls for a very cautious and thorough approach, prioritizing research and understanding the risks fully.
So as you've been listening to all of this, what questions are running through your mind? What further information would you want to see before making a decision about releasing OX5034 in Queensland? Think about the balance between finding new solutions to public health problems and the importance of being cautious when we're dealing with complex ecological systems and potential environmental and health consequences. Thanks for joining us for this deep dive.